right, guys, let's talk about this paper. SARS-CoV-2 spike impairs DNA damage repair and inhibits VDJ recombination in vitro. This is a recently published paper by two scientists out of Sweden, and it's been getting a lot of attention on social media, and it's pretty obvious to show why, because this paper states that the spike protein may impair DNA repair mechanisms and inhibits VDJ recombination. VDJ recombination is a mechanism by which our body produces all the different kinds of antibodies that it can to recognize all the antigens. So it's very important. Without VDJ recombination, we would have non-functional adaptive immune systems. So a spike protein that inhibits those things is a serious problem. And then it gets a lot of press on social media since obviously all the current vaccines are essentially full-length spike proteins. All right, so let's go over what they did, and let's just skip right to the results. And what they did here, okay, so first of all, effect of nuclear localized protein. So what they did is the first thing they did is they constructed a spike protein into a plasmid and put a histidine tail on it. So that means they added like usually 6 histidines, which are amino acids, on the back of the spike protein so that they can track it very easily. And so they did that, and they'd used a bunch of NSP proteins from the virus as well. Those are known to go to the nucleus, and they also found the spike protein. And they said we confirmed their expression and localization by immunoblotting immunofluorescence. Our results were consistent with those from previous studies. They referenced this study, which is systemic and molecular study of subcellular localization. That is this study right here. And they say that what they found is the NSP 1, 5, 9, 13, and 14, and 16 are indeed localized in the nucleus, and nucleoproteins are mainly localized in the cytosol. Surprisingly, they found the abundance of spike protein in the nucleus. So they're saying, we found that these NSP proteins localized in the nucleus, that's the same as the previous studies. And then also they found spike proteins in the nucleus, and they said that was consistent with previous studies. However, if you look at this study, this is systemic and molecular study of subcellular localization. This is the study that they reference. This was published in Nature back in May of 2021. If you look at that, they'll say, as seen in supplementary figure 2, the viral proteins are either cytoplasmic, that means not in the nucleus, and those are these NSP proteins, and the spike right here. So this paper says that the spike protein localized to the cytosol, or both nuclear and cytoplasmic, and the spike protein is not in here. So in this previous study, they found that the spike protein is in the cytosol and not the nucleus. However, in this study, they found that it is in the nucleus as well as the cytosol. But they said that it was consistent with previous studies, which is not true. It's consistent for the NSP, but not for the spike. And the spike is what we're interested in this case, right? Usually in cases where your findings are different than a previous finding, you should discuss that in your discussion and say, here's the reasons that we think that we have a different result than the previous people. However, they said that it's consistent, and they did not they didn't kind of rectify the differences in their discussion because they had said it's consistent, although that isn't the case. But anyways, they go through here, and basically, if you look at this study, they use a DAPI stain. The DAPI stain stains the nucleus. So here's the nucleus, and then this is the histidine tag on the proteins that they made. So they made all these proteins and put a histidine tag on the back end of it, and then those you see where it localizes. Now what you see is the nucleocapsid, which is kind of the shell of the virus, the protein that makes the shell of the virus. You can see that it's in the cytosol. It's not in the nucleus, it's outside the nucleus. So see, when they fuse it together, you see that the blue is the nucleus and the green is the protein. The green is not in the nucleus. However, all these other proteins, the green overlaps with the blue, so they're in the nucleus. So this shows that the spike and the NSP proteins are in the nucleus, and the nucleocapsid is not in the nucleus. What are some possible reasons for that? I mean, one way that's possible is these people are right and the previous people are wrong. In those cases, you kind of need like a third study to do a tiebreaker, and or you need multiple studies that kind of show one to show which one is the outlier. But if you just have two studies and one study shows that it is in the nucleus and one study says it's in the cytoplasm, you kind of don't know which way to go on that. Now there is another paper. This is an old paper from 2004, and they said intracellular targeting signals contribute to the localization of spike protein to the virus assembly site. This paper says the spike protein is membrane bound and also in the cytosol. Now that kind of makes sense if you think about it, because the spike protein ends up on the outside of the virus, and the virus is an enveloped shell with the nucleocapsid in it. So the spike protein has to be stuck in the membrane of the virus for it to work. So you would expect that the spike protein should be in the cytosol or membrane bound, and that's what this paper shows. So these two papers are kind of consistent, and this paper is kind of the outlier. However, there's only three papers, so you kind of got to wait for more data to conclusively say one way or the other. The other possibility is maybe this histidine tag changed its localization. That wouldn't be likely, because the localization of a protein is determined usually by its signal peptide, which is on the front of the peptide or the N terminus. This histidine tag is on the back of the protein or the C terminus, and usually that doesn't affect where it goes. That's not where there's a thing called a signal peptide that's kind of like the address. It tells the protein this is where you need to go, and the his tag is on the end. So I don't think that's one of the issues, but it theoretically could be the issue. Anyways, this first figure shows that the spike protein localizes to the nucleus. So what does that mean? The nucleus is where all the DNA is. It's where all the DNA copying and repair and all that stuff happens. So if the spike protein's not in the nucleus, well, there's no way that it could affect that really. But this showing that the spike protein is in the nucleus opens up that possibility. All right, so let's go to the next repair. So in this next thing, they basically look at how efficiently it can repair a DNA strand, double strand break. So remember, DNA is a double helix. It's like a ladder. It's got two chains, one on each side. A double strand break is when both strands are broken. And that happens very often with like radiation exposure or some kind of environmental toxins. And you have to be able to put that back together. And there's many mechanisms by which we put that together. And what this shows is this is this is how it can be repaired. It can be repaired by what's called homologous recombination, or it can be repaired by non-homologous recombination. And so you see here that the full-length spike protein decreases the efficiency to like 25% or 33%. This first thing is EV stands for empty vector, okay? So that means they just put the plasmid in, but it's not encoding any protein. And that's the way, most studies ideally should be done with what we call a negative control and a positive control. Now, the negative control in this study is the empty vector. So they're saying they don't expect the empty vector to do anything, and so they're going to compare everything to the empty vector. And what they show is the full-length spike protein is different from the empty vector. Now, I don't know why they decided to use an empty vector. A lot of times you can use a kind of a very common protein, like something like albumin or something, and have it express a non-specific common protein like albumin. And that might have been a little better because it's actually producing something. You can say this thing that it's producing didn't cause any problems. With an empty vector control, you could make the argument that how do you know that it producing anything would have disrupted it? And that's possible. The one argument against that is that they had it produce the S1 subunit, and that did not have a big effect. So this does seem like it's some kind of more specific effect than that. Then they tested it by using a lot of kind of DNA damaging things, such as irradiation, doxorubicin, and free radicals, hydrogen peroxide, oxygen free radicals. And these all cause DNA damage. And you can see that the spike protein makes it so that it takes on more damage from those things. So, so the body is not, the cells are not able to repair the damage done by these environmental mutagens as effectively. They ran this test on what's called 282 cells. These are kidneys. This is a embryonic kidney cell line that was from an aborted fetus. And so these are kidney cells that they're running it in, which probably doesn't make a difference. It's probably just as valid because all of our cells have DNA repair mechanisms. Okay, next they show, basically this slide shows, again, they see that the histidine tag is in the soluble nuclear fraction and the chromatin bound fraction, the spike protein is in there. And they show that it keeps BRCA from being recruited into the nucleus as well. So see with the empty vector, you see the BRCA is in the nucleus quite well. And then with the spike protein, it's not in there quite as well. And so BRCA is one of these nuclear repair, is involved in nuclear repair of DNA double strand breaks, and so it's not being localized as well. So far we have the spike protein localizes in the nucleus. The spike protein seems to reduce efficiency of DNA repair, and then the spike protein seems to keep the proteins that are necessary for this kind of out of the nucleus, kind of competes out of it. And then the last thing they show is this, that it impairs VDJ recombination. So you see this is the empty vector, and then there's about a 50% decrease in VDJ recombination. And like I said, VDJ recombination is very important for generating diversity. The way it works is, let's see here. I think one of these had a pretty good. Okay, here we go. This one, so the way it works is there's something called a variable domain, a diversity domain, and a joining domain, and a constant domain in the antibodies. And this VDJ domain, there's many different like pieces you can plug in, and you can combine like this piece with this piece and this piece, and depending on the combination, the conformation is different. So because there's so many different um, subunits that you can choose from, it's like a combination lock. So you can have many different combinations, and because of all these combinations, you can generate millions and millions of different types of antibodies. And then if you add somatic hypermutation into the mix, you can have billions of different kinds of antibodies. And so this is very important. And the way that this works is your body induces a double strand break using this protein called RAG, and then it repairs them by like putting them together. And so that's how, so your body induces double strand breaks and then puts them together, and that's how it generates this diversity. And it's showing that this recombination efficiency is much less in the presence of spike protein. So that's bad because you need VDJ recombination for you to make lymphocytes that recognize things. So this is their discussion, and this is the part where they should have talked about kind of when they had some differing data from previous studies, but they thought that their data was the same as the previous study, so they don't explain the discrepancy. One thing that they do mention is our findings provide evidence of spike protein hijacking DNA damage repair machinery and adaptive immune machinery in vitro. We propose a potential mechanism by which spike proteins may impair adaptive immunity by inhibiting DNA damage repair. Although no evidence has been published that SARS-CoV-2 can infect thymocytes or bone marrow lymphoid cells, our VDJ reporter assay shows that the spike protein intensely impeded VDJ recombination. So what it means is VDJ recombination happens when precursor lymphocytes are being made, and there's only two places in the body that happens. That's in the thymus for T cells and bone marrow for B cells. And B cells are not called B cells because of bone marrow, although a lot of people think that. It actually is called B cells because they were found in the bursa fabricius, which is this organ in chickens. And the original guy that found B cells found it in chickens. Anyways, um, you have to have it infect thymocytes or bone marrow lymphoid precursors for it to affect VDJ recombination. VDJ recombination doesn't happen like in your lungs or in your throat or in your nose or anything like that. So, or in your lymph nodes even. Um, by the time the lymphocytes get to your lymph nodes, they've already had undergone this VDJ recombination, and they are just kind of immature cells that are waiting for the signal that they're the antigen that they can recognize to show up, so that they can mature. But they're saying they know of no evidence that it actually infects these. So if SARS-CoV-2 does not infect these, or the spike protein is not expressed in these cells, it really doesn't matter for in terms of VDJ recombination. All right, then this is them talking about how older people seem to get it worse, and maybe that has to do with this VDJ recombination effect. So this is discussion is where the authors can kind of theorize what they're talking about. Okay, so then now that we know what they did, so let's recapitulate what they did. They found the spike protein goes into the nucleus. They found that it decreases efficiency of DNA repair. It decreases efficiency of localizing the important proteins for DNA repair into the nucleus, and then it reduces VDJ recombination efficiency. So those are the four things that they discovered in their paper. What are the contexts that we need to know about this? One is this has not been replicated. It's one study, and so for for something like this, it probably should be replicated, especially since some of their data does not correlate with previous papers. These two previous papers showed that the spike protein does not go in the nucleus, and their study shows that it does go in the nucleus. So we need to kind of figure out that discrepancy. Um, in terms of a mechanistic thing, the spike protein, like we talked about, is on the surface of the virus, and you would expect it to be in the cytosol or membrane-bound component. You would not expect it to go into the nucleus. So that from the way that we understand the virus works, these other two papers seem to make a little more sense than this newer paper. But we don't know who's right at this point. Um, the other thing is a lot of people have used this paper to say, well, you know, the vaccines use full-length spike protein, and the full-length spike protein could be causing all these effects. Now, there's a couple of things that we need to understand about that. One is that the spike proteins used in the vaccines are not the native spike protein. They have a proline substitution in the center that keeps the spike protein from bending and working as it should. It keeps it in its pre-bound conformation. So we don't know if the vaccine spike protein that has the proline substitution would behave the same way. That would be nice if anyone does a follow-up study. It would be nice to run a study where you use the proline substituted spike protein and see if you get the same effect. The other thing that would have been nice to do is there are coronaviruses that have been around for a long time, and there's four of them actually that cause the common cold, and they all have spike proteins, and we should see it would have been nice to run the same assays with a spike protein from one of those viruses, because if you get the same result with those spike proteins, then we know that this whole thing is probably not that relevant in an actual person, because the implications here are is if the spike protein is doing this, the potential problems are that if you can suppress DNA repair, you're more likely to get things like cancer and other genomic derangements, which are not good. And then two, that you would suppress your adaptive immune system. Now, if a similar assay running it on the spike protein of one of these coronaviruses that have been around for decades, giving us the common cold, show the same thing, then we know it's not a big deal because we've all been getting the common cold. We've all been infected with these other coronaviruses before and been exposed to spike proteins that theoretically do the same thing, and it hasn't been an issue. So ideally, if you wanted to run this study again, it would be nice to run it with the spike protein of one of the common cold coronaviruses, as well as the spike protein with the proline substitution to see if it gets the same thing. Um, so other than that, I think it's a very interesting study, and someone should repeat it because there are some implications for this. The other thing to note is that for people that are worried about the vaccine causing these problems, understand that um, if you get COVID itself, you are going to get the spike protein. And not only are you going to get the spike protein, you're going to get all these NSP proteins as well. And they all also suppress DNA repair according to this paper. So not only will you get the spike protein, you'll get everything else as well. So you'll get probably even more suppression of the DNA repair if what they're showing in these embryonic kidney cells in vitro happens in a person in vivo, which we don't know. A lot of things don't translate from in vitro to in vivo. Um, but if you get the actual coronavirus, you will be exposed to all these. And then we also don't know, these are not all the proteins that the coronavirus encodes. There may be more, and if they assayed those, some of them might also suppress DNA repair even more. So that's what we don't know. So just understand that if you get the vaccine, you're getting just this. Um, and then we also don't know if the proline substituted spike protein would give you the same effect. Possibly it could be worse or it could be better. We don't know because no one's tested it. But you will also be getting these if you get the actual coronavirus. And these also cause suppression of the efficiency of double-stranded DNA breaks. All right. So I hope that gives you a little bit of context about this paper. It should be repeated and something that should not be ignored. Their suggestion in the end of their paper was that maybe we should use not the full-length spike protein, but subunits of the spike protein, and that's why from the very beginning, I've been kind of interested in the Novavax vaccine. That is a protein subunit vaccine. So that should theoretically not have these concerns. My suspicion is that if they looked at the spike protein from one of these cold viruses, they would get very similar effects from this. And then if that were the case, I wouldn't be too concerned about this. If if someone repeats a study with the spike protein from one of those cold viruses, and that spike protein does not have this effect, and it's only SARS-CoV-2 that has effect, then this is pretty concerning actually. So then this may mean that this is some biologically relevant effect, and we should be worried about the effects of the spike protein on DNA repair. Okay, well, thanks for watching. If you have any questions, comment below.